welcome to episode 13 of Divine Superconductor Radio. This is your host, Matt Blackburn, and today we're speaking with Kiran Krishnan, microbiologist and formulator of Just Thrive probiotic and antioxidant. The probiotic field is really interesting because there's so much misinformation, and this interview clarified a lot for me. We just broke down so many myths about strains and the more the better and refrigeration, and there's so much quackery in the probiotic industry. It's really fascinating. So that's why this episode's called Why Everything You Know About Probiotics is Wrong, because we go into it, and this guy is a wealth of knowledge. Talk about what are the most important strains. We talk about leaky gut syndrome. We talk about taking probiotics on an empty stomach and whether that's smart to do. Glyphosate, what kind of nutrients probiotics can make. And by far my favorite part is talking about endotoxin or lipopolysaccharides. A lot of people have never even heard of these terms, and these can contribute to a lot of things: fibrosis, to osteoarthritis, depression, even cancer. Really interesting. They're basically potentiators of almost every chronic condition. So you guys are really going to dig this one. Enjoy. All right, I'm here with Kiran Krishnan. He is a microbiologist and the formulator of Just Thrive. Welcome to the show.
Thank you so much for having me. It's good to be here.
Yeah, I'm really excited. I've been fascinated with probiotics for years and done all sorts of crazy experiments with sauerkraut and kefir and using colostrum and yogurt and all this different stuff. And there's all sorts of different beliefs about probiotics. And I'm connected with a lot of people that hate supplements and they say probiotics don't work, or, you know, they can do more harm than good, or they do nothing. And there's just so many opinions out there. So I'm really excited to speak with you and really break this down.
Yeah. And of course, it's a tsunami of information, and I would say misinformation as well. And there's no doubt that a lot of those people are right in many ways, that probiotics in general, what you have access to, don't really do much, you know, and some of them can cause more problems. And a lot of it comes down to the fact that we really didn't understand anything about the gut and the microbiome until about 2009, 2010. You know, this is all completely new science. We didn't even know what was living in our gut and how many species there were and how they impacted our health and wellness, what they did. We didn't know any of that stuff until some of the initial studies from the Human Microbiome Project started coming out. And that, again, wasn't until about 2009, 2010. You know, so we're only about 8-9 years into this. And our strongest part of our understanding really has only been in the last 5 or 6 years. And, you know, the vast majority of probiotics that people have access to were formulated well before we knew anything about the microbiome. And so a lot of it is based on assumptions. And a lot of it was based on, you know, just kind of old folklore and ideas. And now that we have a better understanding of the gut, we have to evolve our understanding and use of probiotics as well.
That's well said. Yeah, that reminds me of the omega-3 thing and MERS in the early 1920s. B6, zinc, selenium, a lot of the B vitamins and the trace minerals weren't discovered until the 1940s with the University of Washington. And so now we have this concept, this myth of essential fatty acids, where they're not essential. They just create B deficiency, zinc and selenium. So I think this is kind of a similar thing with the microbiome. Is we're just, like you said, we're 2009. That's super recent.
It's very recent. Yeah. And our understanding is changing almost every year, right? Because as we dig more into the microbiome, we find that, you know, we don't even know what we don't know, because the science is so vast. You know, we've known more about bacteria at the bottom of the ocean, you know, 10,000 feet below the surface of the water than we've known about what's growing in between our mouth and our rectum. You know, so it's an unexplored territory. And so, you know, the presumptions that are made in our industry about what can help the gut, what should be in there, like the whole idea of what I call kitchen sink formulas, right? So these are the probiotic products that have 15, 20, 30 different strains in it, and kind of these arbitrary dosages of 50 billion, 100 billion, 200 billion. All those things were all developed in a vacuum of information. We had no idea what anything was happening in the gut, and then people were putting together these products and putting them on the marketplace. It's really fascinating to see. And so, one of my passions and drives is to start dispelling some of these old ideas, and let's move it to modern science. If I'm a company that's been selling a probiotic product since the 1990s, and it's 100 billion CFUs with 17 strains in it, you should have the integrity and the courage to go, yeah, maybe this product wasn't the best fit, and let's reformulate it and change it now based on our modern understanding of the microbiome, and provide people with something that's effective. And so that's where a lot of my drive and passion comes from, is educating people on this topic, because this is our active area of research.
I love it. Yeah, we're going to be breaking down a lot of myths in this episode about probiotics, and that's how I got into it. Like you mentioned, 17 strains. My understanding, even a few years ago, was the more strains, the better, like 30 plus strains, and the more CFU, the better, like getting up into 100 billion, 200 billion, whatever. And so I guess we can start there with the probiotic count. That's one of the biggest myths, right? The more, the better. Like 50 billion is better than 4 billion or something like that.
Yeah, exactly. And that became, it's part of our megalomaniacal society that we live in, right? Everything is bigger. Bigger is better. Supersize everything. Bigger cars, bigger tires, bigger trucks. Everything needs to be bigger. And it's not true when it comes to the microbiome. There's actually no studies that show that 100 billion is any better than 50 billion, or that 50 billion is better than 2 or 3 billion. The dosing response is just not there in the microbiome. It really comes down to the strain and what that strain can or cannot do within the microbiome. And the whole idea of just inundating the microbiome with what we call quote unquote good bacteria to create balance is also out the window. We know that even if the probiotic product has 50 strains in it, that is a fraction of a percent compared to what is actually living in your gut, right? So in a relatively healthy gut, we would have anywhere from 600, 700, 800 to 1,000 species of bacteria, and there's 100 trillion cells, bacterial cells, in the system. So even if you were putting in 100 billion, as a way of creating like a volumetric change in the microbiome, you would be putting literally like a drop of ink into the ocean. 100 billion, as much as it sounds, is nothing compared to 100 trillion, which is what the sea of microbes are in the gut. And even if your probiotic has 50 strains, you're putting it into a population that has up to 1,000 strains. Those 50 strains are not going to have an impact just from a volumetric scale. So most of the probiotic ideology came from this volumetric impact that we were trying to create in the gut. So the idea of more is better became the prevalent idea. If 50 billion is good, 250 billion's got to be better. That's the ideology behind it. And now we know that it's all nonsense. And then the biggest thing of it all is the vast majority of it is dying in your stomach anyway. So really, when you take a 200 billion CFU product or a 50 billion CFU product over 2 or 3 or 4 or 5 billion CFU product, all you're doing is just adding a higher number of dead bacteria moving through your system, and then you're pooping them out 12 hours later. So and then and I'm sure we're going to talk about this with the whole refrigeration thing. So it's just there's a lot of those myths out there, and we come to know now that more is not better, and it's all about the strain. So it's the quality and the specificity of the strain and what it can actually do in the microbiome over just adding in tons and tons of bacteria.
Yeah, you just said a lot of awesome stuff, and I want to dive into a lot of areas of that. The more strains, the better thing. I mean, I used to recommend, I live in the mountains, and going to high elevation to a forest and just, getting wild dirt, high elevation under your fingernails. That's a great way to inoculate with different species. But as you said, you're not going to add new species of probiotics of good bacteria just from a pill.
Right.
That really happens in nature, right?
Exactly. Yeah. Really what we're looking for is effector strains, strains that have some sort of influence or effect on the rest of the microbiome. Like for example, take an influencer, if you will, right? And something that everyone can relate to, or whether it's a political leader or a celebrity or something like that. This individual can influence millions of people by creating and affecting change by appealing to the hearts and minds of the people, right? That's the same thing in the microbiome. There are species that have the capability of influencing intrinsic change within other microorganisms without the brute force of volume. And we always had this mindset of brute force of volume. That 500 billion is actually a big number of good bacteria we're throwing in, and that's going to, overthrow the bad bacteria. In fact, it doesn't do that at all. That volume makes no difference. You need the quality of the strains, right? So we need the appropriate influencers to be able to go in and affect change in the rest of the microbiome. One of the things you brought up, the dirt, is one of the biggest influencers in our natural microbiome.
Awesome. So what are the most important strains? Because in Just Thrive, is it 4 strains?
It's 4, yeah. 4 Bacillus endospores.
Yeah, and that's the biggest differentiator is the strain and that they're spore based. What does spore based mean?
Yeah, so that's an important question. Now, people have often asked me, what is the difference between that and regular bacteria? And then they often conflate that soil organisms are all spore based organisms. So it's important to clarify that as well. So when an organism is a spore-based organism, it has a very specialized capability of going into the spore conformation, meaning it's not metabolically active. So if people remember bacteria, they multiply by something called binary fission, right? So 1 cell becomes 2, 2 cells becomes 4, 4 becomes 8, and so on. So they have logarithmic growth. And when they're metabolically active, they're bringing in nutrient compounds into the cell. They're producing nutrient compounds that they spit out into the extracellular matrix, and they're multiplying rapidly. That's a metabolically active bacteria. When they're not metabolically active, they're dormant. They're what we call in a stationary phase. They can just sit there as cells and turn off all of their machinery. Now, most bacteria will just sit there as a live bacteria cell, and they're very susceptible to things that can kill them. The spores are really interesting in that when they're not in the body, which is their normal environment, they become dormant, meaning they're turning off all of their machinery. And in order to protect themselves so that they can survive outside of the body, they can actually wrap themselves with a protein calcified coating that acts like an outer armor, a shell, that protects them from things that would normally kill other bacteria. In the outside environment, that will be things like UV radiation from the sun, or desiccation in the dry environments, like in the soil when it's dry, or sitting on rocks or plants. In periods of lack of rain, a bacteria will die very quickly if it gets really dry. Compounds that are found like ozone and reactive oxygen species, all these things can kind of kill off bacteria. Certain types of oleoresins and compounds in plants can kill off bacteria. So all of those things can be problematic to bacteria that are in the outside world. And if it's not a bacteria that's designed to naturally exist in the outside world, it can be killed off by these things, especially if it's a gut bacteria. So the vast majority of bacteria that live in your gut, when you pull them out of the gut, let's say in a fecal sample, for example, within seconds, most of them die. Oxygen in itself is that toxic to the vast majority of bacteria in your gut. Less than 1% of bacteria that live in your gut are culturable, meaning you can grow them in a petri dish in a lab. You can't. Most of them are so specialized that when they're outside of the gut environment, they almost immediately die. So these are very specialized type of bacteria that have been designed by evolution to exist in the gut alone. The spores are so interesting in that they're so specialized that they can live in the gut in that one very unique environment. But because they can form the spore, they can also leave the gut through defecation and exist in the outside world for an indefinite amount of time. The oldest spore that's been found that's still alive is 250 million years old. They found spores that are 50 million years old, fossilized in amber, that they can plate still and grow it. It's that old and it's still alive because it's sitting in this spore form. And then the spore form also allows it to survive through your gastric system, which is the stomach acid and the bile salts, which kill off the vast majority of living organisms that go through your digestive system. And they can survive that and get back to their home, back to the intestines, where they then come out of this spore shape, this spore conformation, and become a live probiotic cell again. So imagine, you mentioned the dirt thing. This is really interesting to think about. So if you go to upper elevations where the environment is fairly undisturbed by the human population, you'll find spores sitting there in the soil that could be 2 or 3 million years old, right? And haven't really done anything in that 2 or 3 million year period. Then you get it in your fingernails, and because you're smart not to sterilize your hands before you eat your sandwich, you'll get a couple of those spores into your system. They survive past your gastric system, and they've been dormant for 2 or 3 million years. Within 20 minutes of entering your gut, they'll break open from this spore shell and go to work cleaning up your gut for you. That's how fascinating these organisms are. And that's why we kind of honed in on them, because they are major influencers in the gut.
That's amazing. Yeah, there's a lot there. One thing that came up for me was gut testing. I've done one test. There's a big one in the biohacking community where you send in a fecal sample. So are you saying because only less than 1% are culturable and they die 99% in the presence of oxygen, do you think those are not effective or they're not accurate?
Yeah, so most of those gut tests use DNA or RNA sequencing to test. So the bacteria don't have to be alive in order to be detected. However, that being said, those are still incredibly inaccurate. Most of them use a sequencing process called 16S ribosomal RNA sequencing. And I'm sure most of your listeners have no idea what in the world that means, but I can give you an analogy, right? So it's like, imagine there's a crowd of 1,000 people on a street, and you are at the top of a 50-story building, and you're looking down on that crowd of people, and you're trying to identify the individuals by looking at the top of their heads, right? And so it's that poor of a resolution. They do things, laboratory techniques, to try to improve the specificity, but at best they're about 50% accurate. The other problem with it is that how you sample your stool. So if you look at a solid stool mass, that solid stool in itself is a very complex ecology. So the surface of the stool has completely different types of bacteria than when you penetrate even a couple of millimeters into the stool. So the inside part of the stool has a different ecology than the outside part of the stool. And the vast majority of those tests, get you to swab the surface of the stool and send that in. So you're really picking up a fraction of the microbes that are actually in that stool itself. What you really need to do to get a better sampling of that stool is do a coring sample, where you pull a core through the entire 3D surface of the stool, and then do that in a few different areas of stool. Because the other problem is bacteria is not homogeneous in your stool, right? Any given area of the stool has a slightly different representation of the types of bacteria that are in your microbiota, because bacteria aren't spread evenly throughout your feces. Then the last problem is you're also only measuring bacteria that's being detached and coming out. So it's not representative of what's actually going on in most of your microbiome. So it can be used to give you some sort of clue, to give you some idea, but people use them way too much to try to figure out therapies for their gut, and it can't tell you that. The other thing that drives me crazy about many of these tests is people have sent me thousands of reports over the last few years of their stool tests, and they go, what do I do about this test? And I look at it, and the test is saying Klebsiella is high, or Pseudomonas is high, or E. coli is high. That's complete nonsense. There is no threshold for what is high and what is low on most of these organisms, because everyone's microbiome is completely different. There are certain universal rules within the microbiome, but for the most part, when it comes down to the species level, everyone's microbiome is completely different. Even identical twins who have 100% the same genetics can have up to 50% differences in their microbiome. So to say that this test shows your Klebsiella is high is complete nonsense, because there's no establishment of what's the normal level, what's high and what's low of any of these organisms, right? So that's a problem, because a lot of people get these reports back and they'll look at it and they'll freak out, and then their practitioner or doctor will look at it and go, something's high, we got to treat it with antimicrobials or antibiotics, and then they start hitting the gut with all of these antimicrobials and antibiotics, and more often than not, that leads to more problems than resolution.
That's amazing. Going back to the spore-based thing, what's the difference between enteric coating and spore-based? Because I know that enteric is important for things like systemic enzymes and certain enzyme products, but probiotics are completely different, right?
They're biological functioning units. The whole idea with enteric coating or putting bacteria in special capsules, or even there's some company that wraps them in seaweed and some crazy stuff that people are doing. The whole idea behind all of those is let's somehow engineer the bacteria to try to make it past the stomach acid and the bile salts. And in my view, being a microbiologist, but also always paying attention to evolutionary biology, to me, if the bacteria isn't designed to do something, when you have to engineer it to do it, then you're not following the rules of nature, how things are intended to work. And so enteric coating a bacteria basically means taking a bacteria that is not designed for a particular function and then trying to get it to function that way by getting it past the stomach acid. Now, as it turns out, we've actually studied numerous products with enteric coating, and all of the special capsules and all that. Even with the special capsules and all that, they do not survive through the gastric system. At a pH of 1.7, 1.8, 1.9, which are normal, healthy physiological pHs in the stomach, they don't survive. Even the capsules can't resist that. So what companies will do, they'll say, we've verified it to survive the stomach acid. And when you look at the study, they've done the study at a pH of 2.8 or 3, which is at the very alkaline end of where your gastric system would and should be. And then, and if people don't understand, there's a huge difference between a pH of 2.8 and 1.8, because the pH scale is a logarithmic scale. So it's a 10-point scale. So each point is equivalent to 10 points, right? So a pH of 1.8 is 10 times more acidic than a pH of 2.8. So if you show that your enteric-coated probiotic survives at a pH of 2.8, it doesn't mean by any stretch of the imagination it can actually survive in the normal gastric system. So anytime anyone has had to do anything fancy to the bacteria in order to try to make it act as a probiotic, it's more than likely not going to function as a probiotic in the body.
That's a really good point. Maybe they're drinking too much alkaline water, right?
Exactly. Right. Yes. And taking too much snake oil at the same
time. So I know with a lot of supplement companies, they don't have to, like, legally, they can get away with a lot putting whatever on the label. So as far as the strains and the potency of probiotic products, how strict are the label laws? Are they pretty relaxed where they can kind of just say whatever or not really back it?
Yeah, I mean, a lot of it, and the industry came up with this interesting nuance to get around labeling issues. So the regulatory part of the industry, the FDA doesn't really regulate product labels very much. Certainly not supplement product labels. When it comes to things like CFU count, strain number, and all that stuff, that rarely does the FDA, pull product off the shelf and check that stuff. So it's all self-regulation within the industry. The biggest regulators of that are the class action lawyers, right? So there's a whole group of lawyers who have found these opportunities to find companies that are mislabeling product. And then because most of these products are sold across state lines and there's thousands of consumers that have bought it, then they can create a class and file a big, massive class action lawsuit. So those have actually been the biggest regulators of the supplement industry, even when it comes to probiotics. Like, for example, Dannon yogurt had to settle like a $30 million class action suit. And then the FTC got involved in that one as well. once these lawyers brought up the issue. But a lot of it is on mislabeling of the probiotic strains and survivability and functionality of the strains. So the FDA does not regulate it. So companies, unless they get really big, they don't really care about the regulation as much as they probably should. Now, when it comes to CFU count and strain count, one of the tricks that the industry developed is when you say a product has 50 billion CFUs, now they cannot guarantee that the product still has 50 billion CFUs at the end of the shelf life of that product or at the point that the consumer consumes the product. So you'll see on a lot of probiotic products, what they've done is they've changed the labeling where they say 50 billion CFUs at time of manufacturing, right? That is a dirty little trick that the industry developed to say, we don't know where the count is going to be 3 months, 6 months, 12 months after production, but we know right now we're putting 50 billion in, so that's all we're claiming, is that's what we're putting in. The CFU count could be 0 at the end of 3 months after production, and they don't care about it. All they care about is saying, we're covering our butts because we're saying we put in 50 billion at manufacturing in case one of these class action lawyers grabs a product off the shelf and shows that actually there isn't 50 billion, it's only 10 billion, then sues them. They can say, no, our label claim is 50 billion at manufacturing and we're covering that. So and to me, this whole thing is devoid of science, right? The fact that you even had to come out with a alternate labeling claim at manufacturing means you're not doing anything based on science, because who in the hell cares what dose you're putting in at manufacturing? What you should care about is what is the dose that the consumer who's spending their hard-earned money buying the product, what dose are they getting? And then is that dose going to be effective? So there's a lot of nuance in that, and there's almost no value in looking at the probiotic label at all in most of these cases.
That's a great segue, because when I got into probiotics, I had the thought that, refrigerated is better, and I'd go to the health food store and, there's maybe 30, 40 probiotic supplements in the refrigerated section, inside of the supplement section. And I thought that was superior to the room temperature ones. So that's a whole nother myth, right? Do probiotics need to be refrigerated, or what's the deal with that?
Yeah, so that's I'm glad you brought that up, because when I started researching probiotics, and I was taking the scientific approach to it, but the way I started was actually just to go survey the market. And I would go to health food stores and I ask them, what are your best probiotics? And the response I always got was the stuff in the refrigerator. Those were the best probiotics. And my question to them always was, why are they the best? Why are you saying it's the best? Why are you recommending these? And they say, well, because these are live culture. And it's really important to have a live culture in your probiotic. So we keep it refrigerated to keep it alive. And they would always say, when you get it home, make sure you quickly get it into your home and put it in the refrigerator. So you don't want it sitting in your car or in a shopping bag for too long, and so on. And then I'd ask them, so, okay, if it sat at room temperature at 70 degrees, these strains would die over time. And they said, yes, that's why you got to keep them cold. And so then my question always was, well, if they can't survive at 70 degrees at room temperature, it's 98.6 degrees in the body and a pH of 1. How are they going to survive in that? And, no one ever had an answer for me. And I would be that annoying guy calling the 800 numbers of many of these companies going, how do you justify a refrigerated probiotic that can't survive at room temperature and claiming that it has any function in the body at all at 98.6 degrees? And nobody had any response to that. I never once got a company that gave me a report or study or anything that said, here's our justification for that. So my immediate thought was, any of this stuff in the refrigerator is complete nonsense. If it has to be refrigerated, there's no way it's functioning as a live probiotic in your gut. And in fact, then you dig into the industry a little bit more, and I have the fortune of being behind the curtain in the industry in many ways. And you start to find out that, again, a lot of this is because they're afraid of certain legal repercussions, because one of the ways that you measure CFU count is through plating, right? Which it requires live bacteria. And so in order to be able to take the product off the shelf and then plate, which means you're growing the bacteria in petri dishes, to figure out the count of bacteria in there, the bacteria have to be alive in the bottle. And so companies started to become scared that if they kept it on the shelf and the bacteria started to die off, that at some point somebody's going to test it once it's 2 or 3 months old and find that it has a fraction of the live organisms that it did when it started. And so they started saying, we got to ship these under refrigeration. You got to put them in the refrigerator because they want to maintain the live culture. And then that way, if somebody tests the product and shows that it has a fraction of the live bacterial culture in the product, they can point to a disruption in the cold chain supply. They can say, well, we said you have to refrigerate it. It says right on the bottle, on the label, you got to refrigerate it. But when this distributor shipped it, they had 24 hours of temp abuse and it wasn't in a refrigerated truck, or when this lawyer picked it up, how do we know they maintain the cold temperature? So they have some legs to stand on, if they get in trouble. So it's again devoid of science. Complete nonsense. To me, if a product has to be refrigerated, it's not going to work as a probiotic. So you could skip the refrigeration. And where did our ancestors get their refrigerated probiotics from, right? Where in the course of human evolution were our ancestors going, oh, here's bacteria that's refrigerated and I'm going to take them in between meals precisely and get my dose of bacteria in. That didn't occur in nature. So it doesn't make any sense at all.
Interesting. So I'm curious, let's say I'm on a camping trip or I'm hiking and say it's over 100 degrees. Mm-hmm. Should I keep, and I have some Just Thrive probiotic with me, should I keep it in a cooler or is it relatively safe around 100?
Oh, we've in fact baked the spores. We baked them at 455 Fahrenheit for 8 to 12 minutes and they're still good. So they're extremely robust. Yeah, no, you can leave them if you live in Arizona and it's 125 degrees in your car, you can leave them in your car. The bottle might melt completely, but the spores will be perfectly fine. Remember, these are organisms that can survive in the outside environment indefinitely for millions of years. In fact, there's a study on Bacillus subtilis, which is one of the strains that we have in our product, where they were looking for organisms that exist on Earth today that could have seeded Earth with the preliminary, building blocks of life, nucleic acids and protein, amino acids, and so on. There's a theory called panspermia. I don't know if you're familiar with it, right? So the idea is that meteorites, that were bombarding the Earth in early Earth's history, brought with it nucleic acids and other components of living organisms that seeded what would end up being the first life forms on Earth. So they were testing bacteria that exist on Earth today that could survive 6-7 years in interstellar travel on a rock. And they found that these spores, like Bacillus subtilis, can in fact survive interstellar environments for up to 7 years. And so, these same spores could have very well been the initial seeds of life on our planet. And then they're also a normal commensal part of our gut. And so they're really, really important to how we function biologically. And what we've done is we've kind of built these barriers between us and them over the last few decades, where our environments, our living environments, our food sources, our work environments are all completely sterile, right? And we don't have any more interaction with these species. And that's where we're starting to see so much dysfunction in our microbiome.
Wow. And while we have those sterile environments, we're exposed to countless synthetic toxins, right? Like I want to talk about like glyphosate and leaky gut. That's a big thing right now. And I've been looking into lipopolysaccharides or endotoxin. And that was what really blew my mind about the spore-based probiotics that you guys provide is there's actually a published study, there's a 42% reduction, right, in endotoxin.
Yeah, and that's when you look at the treatment group alone, when you compare it to the placebo group, and the placebo group actually had a 32% increase in their amount of endotoxin that they're absorbing. And that's leaky gut. So for your listeners who aren't familiar, these endotoxins, it's called an endotoxin because it's a toxin that's generated within your own system. An exotoxin would be one that we pick up from the outside. But the endotoxin is generated within the microbiome itself. And so you can't get away from it. And it's a very severe, potent toxin that creates chronic low-grade inflammation, and it sets off the pathology of most chronic illnesses. So there are huge studies from the American Diabetic Association, from the NIH, and all of these major research institutes that show that the presence of this endotoxin in your circulation, which is called endotoxemia, is the major driver of diabetes, heart disease, Alzheimer's, Parkinson's. Most autoimmune conditions are induced by the presence of this endotoxin. Obesity, senile dementia, various forms of cancer that are perpetuated by these endotoxins. So all of the things that we care about as health implications are driven by these endotoxins that are generated in your gut and allowed to leak through your gut lining into your circulatory system. And that's the crux of leaky gut, right? So our whole idea was that you can do a lot of things in medicine and therapy, but if you can't stop the endotoxins that are naturally formed in your gut from leaking through, then everything else you're taking basically 2 steps forward, 1 step back, because the disease source is still there. So our whole focus is if you can use a probiotic that has the ability to reform the gut, both the population of the gut and the structures within the gut, the mucosa and the gut lining, then you should have one that can stop this endotoxemia and this huge increase in LPS endotoxin migrating through the gut lining into the circulatory system. And that's exactly what we found. In the placebo group, as I mentioned, we found a 32% increase in the endotoxemia over a 30-day period. And we saw a 42% decrease in the treatment group, which when you compare the difference between the two, it's over 60% reduction in endotoxemia in that 30-day period. And these are people that didn't do anything else. They didn't change their behavior. Most of them are college students, so they're still doing terrible things for themselves in that 30-day period, being stressed, drinking, eating fast food, partying, all of that stuff. And yet we saw this huge change. Now, it's important to note that even on top of that change, we also saw all of the corresponding inflammatory markers, cytokines, also come down very significantly. And then we saw things like the gut-brain connection being restored from a metabolic perspective. So lots of really exciting things were demonstrated in that study. And now, since then, because we know we can stop this leaky gut and stop this endotoxemia, we are doing studies to see if that same effect is seen in other areas where we know leaky gut has an influence. For example, we just finished a study on a dermatology group where we're seeing that we got about a 45% reduction in acne lesion counts in people with acne in 30 days. We saw a significant reduction in the amount of sebum produced in the sebaceous glands. We saw a significant reduction in inflammation in the skin in that 30-day period. We're doing a periodontal gum disease study right now because there's a deep connection between leaky gut and gum disease. There's also a deep connection between leaky gut and triglyceride levels. And triglyceride levels are quite significant markers of cardiovascular and other disease risk. And what we saw in a 90-day study that we finished was a 48% reduction in triglycerides in that 90-day period. I can tell you that statins, the number one prescribed prescription drugs for triglycerides, lower triglycerides around 15% at best. Then, of course, you've got all of the side effects. Fish oils, the best studied fish oils, lower triglycerides by about 25%. The spores reduce it by about 48%. It's such a significant impact. So we're doing a whole bunch of other studies. Now that we know we can stop this endotoxemia and leaky gut, we're doing a whole bunch of other studies on areas that we know are impacted, like autoimmune disease, for example, obesity. We're just completing a weight management study that is seeing quite significant impact. And then we're going to be doing studies on anxiety, depression, all these other areas as well.
That's incredible. Yeah, I've been just obsessed with researching endotoxin, especially since I learned that spore-based probiotics can really help reduce it. And just diving into the studies, and you mentioned a lot of chronic conditions, but I've seen fibrosis, osteoarthritis, even depression, even HIV can be exacerbated. The most fascinating study, and it's funny you mentioned the college kids, 1998 study I found said that ethanol like alcohol is way more toxic, or perhaps only toxic in the presence of endotoxin. That was really wild to me.
Yeah, absolutely. I mean, because then you think about a couple of different aspects of that. Number one, if your gut is leaky to endotoxins, you're likely getting more of that alcohol, the ethanol compounds leaking through into the circulatory system rather than going through the portal vein and then being cleared by the liver, which is how it's supposed to be, right? So your blood alcohol influence from drinking could be significantly higher when your gut is leaky in the presence of endotoxins. The other thing is when in the presence of endotoxins, your bacteria, your microbiome is off because endotoxins are constantly recruiting inflammatory immune cells into your mucosa. So it's damaging your mucosal layer, it's damaging the microbiome. So the alcohol will have a higher influence on creating dysbiosis and thus toxicity within the microbiome. And the last thing is, when you have a lot of LPS, your liver goes through a lot of stress trying to clear the LPS from your blood and from your system. And so imagine your liver is already processing all of these toxins, and then on top of it has to process alcohol. It makes the alcohol that much more toxic. So then you become more susceptible to fatty liver disease, and then even non-alcoholic fatty liver disease because of the additional toxicity the alcohol is adding. So yeah, absolutely. And we know now that the endotoxins, LPS endotoxin, is a primary driver in the onset of Alzheimer's. It's LPS endotoxins are correlated with just recently, I just saw a study. This is just completed, not published yet. It's going through peer review. How the IgA LPS response dictates the outcome of menstrual cycle for women. The severity of menstrual cycle symptoms is dictated by LPS, by endotoxins. In men, elevated endotoxins reduces testosterone production. So this whole idea of low T and people having hypogonadism, especially in your 30s, 40s, and beyond, is driven by LPS endotoxins. It's so pervasive, it gets into your amygdala, your hippocampus. It screws with the dopamine receptors, serotonin receptors. It causes severe anxiety, depression, all of these things driven by this toxin that's formed in your gut. And we can significantly improve our outcome in these conditions if we can just stop that endotoxin from migrating through. And that's our whole focus of our work.
That's amazing. Yeah, I'm really glad you brought up non-alcoholic fatty liver disease and just the liver in general, because I think a lot of people overlook the liver, and it's so important. And I've been looking at the synergy of toxicity between like lipofuscin pigments, like age pigments, steroid pigments, iron, and LPS. And I think all of that increases estrogen, puts serotonin through the roof, and just really throws off the system. It's like everything kind of working together, I'm finding, that creates these chronic conditions.
Yeah, absolutely.
So what's the difference between glyphosate and LPS endotoxin? Because I was talking to a doctor at the recent expo I was at, and he was saying that glyphosate just punches holes in the gut like crazy. Endotoxin does that, but not to the same degree. Is that true? Or
yeah, so it's different. So glyphosate's impact on the gut is really through the microbiome itself. So glyphosate is an antibiotic. It's a very strange and really scary antibiotic, right? So if you took an antibiotic, like if you went to the doctor and you had strep throat or you had an upper respiratory infection and you were given a clindamycin or Cipro, whatever it may be, those antibiotics basically kill everything in your microbiome at once. So they basically bring down the bacterial population in your microbiome by 99% almost, right? And then the microbiome starts to bounce back after that. The scary thing about glyphosate is it selectively kills good bacteria. It's one of the only compounds that has been shown to actually allow for pathogens, things like Clostridia bolte, tetani, Pseudomonas, Klebsiella, all of these, Salmonella, Campylobacter, all of these pathogens and opportunistic pathogens to do well, and it kills the competitive good commensal bacteria. So over time, being exposed to Roundup and glyphosate through the food source and the environment and the water source, what you're doing is creating this microbiome that is predominant by pathogens and opportunistic pathogens. So it's a really terrible antibiotic. I would much rather take a month's worth of Cipro than being exposed for the next couple of years to Roundup, because the month worth of Cipro is going to really perturb your microbiome, but it affects all bacteria equally, versus the Roundup, which affects only the good bacteria and proliferates the bad bacteria. So the way it creates leakiness in the gut is by creating this severe dysbiosis, which then disrupts the microbes whose job it is to maintain the integrity and structure of the gut lining. So that's how it does it. Now, LPS is through a secondary mechanism, which is where the inflammation associated with LPS migrating through the mucosal layer will cause your own immune cells to disrupt your gut lining, because it's trying to attack the LPS, but inevitably attacks and damages your gut lining cells as well. So, the idea of punching holes in the gut really comes from your intestinal epithelial cells dying and then getting kicked out and then not being replaced quick enough. So you actually have gaps in that intestinal lining. And that can occur with LPS, but it can also occur through glyphosate, because it's causing this dysbiosis.
Wow, that's incredible. I know you have to run pretty soon, so let's go through some questions here real quick.
Yeah.
The empty stomach thing, that blew my mind, because that was the thing that I understood. I thought probiotics were best taken on an empty stomach right before bed, but you guys say no, take them with food, right?
Yeah, and again, back to evolutionary biology. I mean, where did our ancestors take their bacteria carefully in between meals, right? They everything they got from a microbial standpoint, they got when they were eating food, because they ate dirt. They essentially got environmental microbes contaminating the water and food, and that's how they got the bacteria in. The whole idea of using it in between meals is trying to subvert the acid. Right. So because as you start taking the first bites of food, your stomach starts releasing hydrochloric acid at higher amounts. So the whole idea was do it in a fasted state, because then you can try to subvert the acid. But if you had a bacteria that was not damaged by acid, because it's designed by nature to survive, then it doesn't matter. And in fact, we want a probiotic to be coming in with food, because part of the job of some of these bacteria is to help digest the food. Like the spores, for example, make the digestive enzymes in the gut. So they actually help you break down and then ferment the food as well. So yeah, that whole idea of in between meals or an empty stomach at night before bed, it's all, again, just kind of made up stuff in a vacuum of science.
Yeah, it all makes sense logically, everything you're saying. So I don't know why. I guess I was just swept up in all the marketing around probiotics.
Yeah, and the thing is, that's what everyone said, right? So even the most influential people in the functional medicine space, the doctors that write books and all that, everyone just kind of says the same thing, and nobody stops and really looks into it and studies it, because it's coming from multiple sources. That's both the wisdom and the ignorance of a crowd, right? The crowd has certain wisdom to it. But then a crowd also has a certain amount of ignorance, because when a few people are saying it, it becomes the status quo, because people are saying it. And when we dug into it, and it takes a nerdy microbiologist who knows bacteria to really get into this stuff, and that's why I got into it.
I love it. Yeah. One of my mentors, Dr. Ray Peat, says that he calls it the central dogma. Mm-hmm. And he says it's like a religious kind of, dogma in science.
Yeah, absolutely. And when you see it on a Dr. Oz show, you see it in a book or two, and all of a sudden it just becomes the truth, and it's not really validated. It's just an idea that someone came up with and started saying it, and then everyone else repeats it.
Yeah. So you mentioned enzymes. What other nutrients do probiotics create? Because I think on the packaging on Just Thrive, it says they also create antioxidants, right?
Yeah, so that specific strain, there's a strain in there called Bacillus indicus that produces carotenoids. So these are the colored portion of fruits and vegetables. These are really powerful antioxidants, among other functions in the body. So things that people know well are like beta carotene, which most people have heard of, or lutein, things that are really important for eye health and skin health and reducing oxidative stress in the body. So this strain is really interesting because they were studying organisms in the animal kingdom that expressed a lot of these colored carotenoids on their skin. For example, shrimp have those orange bands around them, right? And salmon has a natural pink color to the skin. And in fact, in the salmon market, the value of the fish is determined by how pink the salmon is. Or if you look at flamingos, they have pink on their feathers, and that's part of their mating rituals. The flamingos that are the most pink are selected for by mates as being the most fit to mate and create baby flamingos, if you will. And so how is it that the flamingos become pink? So they eat a brine shrimp, but how is it that the brine shrimp gives them the pink color? All of these animals that express a lot of carotenoids on their skin, which denotes health and wellness in the animal, get it through probiotics. So they don't eat fruits and vegetables, right? So salmon is not eating colored fruits and vegetables. They're getting a spore which lives in their gut and produces these carotenoids at very high levels. Same thing with the flamingos. They're eating these brine shrimp that have these spores in them that then settle in their gut, colonize, and produce these carotenoids at very high levels. Same thing with shrimp and other animals. So the thinking was, and this is a big European research consortium called the Color Spore Research Consortium, over 5.5 million euros went into this study process, and there were about 80 different researchers were involved in figuring this out. So the idea was, if all of these animals in the animal kingdom require their carotenoid intake to come from bacteria in the gut, maybe humans have that same need as well, because they started testing our ability to absorb carotenoids from foods. So if you eat like, a pepper, a red pepper, a green pepper, or a fruit that contains high levels of carotenoids, what is our ability to absorb those carotenoids from food? And then also, what is our ability to absorb purified carotenoids from a supplement standpoint? So taking purified beta carotene as a supplement, what they found was that humans absorb less than 10% of the carotenoids that they consume through foods and through supplements. And so the idea was, because we have such poor ability to absorb carotenoids that are coming in through the diet, we must have bacteria that sit in our gut and actually produce these, or we wouldn't be getting enough carotenoids. And so they did a whole study where they looked at hundreds and hundreds of healthy human volunteers and looked for bacteria in the microbiome that can actually produce carotenoids. And that's what they found. This Bacillus indicus was a normal commensal spore-based bacteria that produces 12 different carotenoids in the gut at RDA levels, so at recommended dietary allowance levels, at the levels you should be getting from your diet. And it produces it right at the site of absorption in the intestinal lining. So it produces things like lycopene, lutein, astaxanthin, zeaxanthin, alpha and beta carotene. So all of these carotenoids that people take as very expensive supplements can be produced as a nutrient factory right in the gut.
That's incredible. Yeah, I was researching astaxanthin for a while, so it's my current belief that supplements are the worst because you can overdose them very easily, like you mentioned, beta carotene. And people are taking high doses of astaxanthin. And then second would be food, but the best is producing them endogenously, because there's an intelligence, because they only release as much as you need, right?
Exactly. Yeah, there's a feedback loop, for all of these things that are being produced. That when you have high amounts, then there's a feedback loop that shuts down the production. And that's the same with like even B vitamins, right? So one of the things we see is that these spores can produce B vitamins, but when you look at the types of B vitamins that they produce the methylated form of the B vitamins, so a lot of people are studying this methylation pathways and the issues. So with the MTHFR mutation, some of the estimates are that the MTHFR mutation is really prevalent. It's upwards of 38% of the population. And in my view, looking at evolutionary biology again, if that gene was so important, it wouldn't be 38% prevalent in terms of mutation, because people would be dying with that mutation, right? Natural selection would have weeded that out. But one of the reasons why it's likely not that important is because your microbiome produces all of the methylated B vitamins you need. And in fact, they produce so much so that less than 30% of the methylated B vitamins produced in the microbiome are actually absorbed. 70% of it actually goes and is converted to other compounds like urolithins, which are that would then go down the cycle of other epigenetic functions. So a lot of the stuff that we need is produced in the microbiome. The other things you mentioned, what other compounds are produced? So the most recent ones we are studying is the ability to produce neurotransmitters in the gut. So the spores can produce serotonin, can produce dopamine, GABA, and there's 4 other neurotransmitters we haven't identified yet, but we know that the microbiome produces all of the important neurotransmitters. The microbiome also produces all of the necessary hormones. Your full endocrine system is produced in the microbiome. And in fact, there's evidence that our endocrine glands learned how to produce these hormones from bacteria, and from the gut bacteria in particular. So they produce, a whole host of things. They're very important for the digestive process. They produce cellulase to break down plant matter. We can't produce cellulase ourselves. So all of the healthy plants and vegetables that we eat can't be utilized at all unless we have adequate gut bacteria to break it down for us and assimilate the nutrients. So really important. I mean, one of the things I always tell people is the old saying, you are what you eat, is completely wrong. It's actually you are what you absorb, right? Yeah. So you can eat the healthiest diet in the world, but if you've got an inflamed, inadequate gut and a dysfunctional microbiome, you're not absorbing and assimilating those nutrients, and you're not getting the benefit of those healthy choices.
Absolutely. Yeah, I used to be raw vegan, and for I think 4 years straight, I ate a big salad, like a whole head of lettuce, like raw beets, raw carrots, all these nuts and seeds. And this was before I understood stomach acid, or the fact that we're not a cow, we don't have 4 stomachs.
Right. Right. Yeah.
And I think that really all that cellulose, I think people are into fiber, but a lot of people are getting too much fiber, and that could be super stressful.
Yeah, absolutely. If you look at our ancestors, right, they were, even though they're called hunter-gatherers, they gathered and foraged way more than they hunted. But they ate meat, and they ate animal products. They ate roots and tubers and seeds and nuts and insects. They ate just a huge variety of things. And when you look at that ancestral diet, you get more and more that the diet was extremely diverse. They ate up to 600 different types of foods on an annual basis. And our biggest problem is we eat a really shrunken diversity of diet. I mean, an average American maybe eats 15 different types of foods, and then if you find one that you like that's quote unquote healthy, then you're going to eat a ton of that. Like, you remember in the early 2000s, everyone was eating soy everything, right? Because soy all of a sudden became the healthy food, and you're eating soy burgers, soy nuggets, soy drinks, soy milk. And then all of a sudden we come to realize that all of that is probably going to increase your risk of breast cancer and other issues. So we overdo these things, and our ancestors never did that. They ate little bits of a lot of different things, and that's how our microbiome is designed to be fed.
I love it. Final question for you. What are we supposed to feel with probiotics? Because I hear from a lot of people, I've been taking probiotics for months. I don't feel anything. That's because they're not surviving the stomach acid, right?
Yeah, exactly. I mean, the vast majority of probiotics you can buy out there are not doing anything, really. So they're probably right in that respect. But if you're looking at the question of, what should you feel, assuming you're taking a probiotic that's working and that is changing your microbiome, that's a really important question, because most people do not relate all these secondary and tertiary issues that they're having to the gut, right? So they feel if they're taking a probiotic, they should be feeling something in the gut, but they may not have that severe of an issue in the gut to begin with. But their issue might be, anxiety, and their issue might be being hard to deal with stress. Their issue might be gum disease. Their issue might be acne. So when you take an effective probiotic that's actually stopping the leaky gut, stopping that LPS endotoxemia, increasing the diversity in your microbiome, increasing short chain fatty acid production, reducing that chronic inflammation. When you're getting all of those benefits, you will see them manifest in many different ways. And it will typically manifest in the issue that you are dealing with as the biggest issue in your time. For example, it may be a weight issue. It may be cravings, just satiety. You're dealing with unusual amount of sugar cravings. One of the things I hear from a lot of people is when they start taking the spores, they're like, I don't know if my bowel movements have changed or not, but I notice that my cravings have gone way down. Or you might notice it in how much better you sleep, or the fact that when you wake up, you're not waking up anxious. anymore. It might reflect in your gums more than anything else, right? So if you're taking a probiotic that's really making a fundamental difference in your microbiome, you have to be very cognizant that the change and the effect can be completely distal or away from your gut. It could be in any other part of your body, and that just speaks to the importance of a healthy microbiome. A healthy microbiome controls virtually every aspect of your biology. And so you can expect changes in places that you wouldn't necessarily see or expect to see outside of the gut.
Yeah, as you mentioned, neurotransmitters, hormones. I think the hormone thing is huge. That some people are doing raw carrot salads or bamboo shoots to actually bind to excess estrogen in the gut and carry it out. And I've had personal experience with that helping. But I think that plus the spore-based probiotic can be huge.
Oh, yeah, absolutely. Like, for example, there's something called estrobolome, which is a constellation of bacteria in your microbiome whose job it is to metabolize estrogen. And if you have low diversity in your microbiome, your estrobolome function is going to be way low, and you're going to end up with estrogen dominance. And that can occur both in men and women. And so one of the ways of combating that is you take the spores. We've shown in a paper we're about to publish in the next couple of months that the spores, those 4 strains entering your system, increases the diversity of your microbiome by almost 45-50%. So it's increasing the growth of all of these other important commensal bacteria, which means your estrobolome is functioning better. You're going to clear estrogen better. And in the case of women, one of the things that they don't necessarily relate to it, there's a cascading effect to that. So what happens when estrogen is metabolized in the gut, right? So one of the reasons why binding the estrogen, taking it out, is not necessarily going to solve the problem, because one of the consequences of metabolizing estrogen in the gut is the estrogen byproducts from the metabolism travel through the mucosa and enter the vaginal mucosa. And in the vaginal mucosa, they become the primary food for the most important bacteria in the vaginal mucosa. So there is this deep connection between the gut and the vaginal tract, where the estrogen metabolism byproducts are supposed to feed the good bacteria in the gut. So not only, if you end up with estrogen dominance, not only can you end up with things like polycystic ovarian disease and breast cancer risk and so on, it also drives a lot of dysbiosis in the vaginal canal, which leads to chronic yeast infections, bacterial vaginosis, UTIs, and so on. So all of these interconnected systems require the function of a proper microbiome. So that, sorry to go into that whole tangent, but that's an important way of seeing how, all of these natural systems are supposed to work and feed off of one another and support one another. And what we want to do to curb our issue is to take the most natural route of fixing it, and in this case with estrogen dominance, it's increasing diversity.
Really well said. Yeah, I get a lot of questions of people trying to sniper a chronic condition. And PCOS, like you mentioned, polycystic ovarian syndrome, seems to be an epidemic right now. It's just not really talked about. A ton of women have it. And it's like, it's not an herb that does it. It's the body, as Josh Rubin says, it's like a system of systems. And so it's really looking at it like that, right? You're affecting the microbiome, which is affecting, like you said, the vaginal canal and everything. So I really loved your analogy too about influencers, because I think looking at Just Thrive and saying, oh, there's only 4 strains in there, how do I know I don't have too many of these? That's probably the question most people ask. But you mentioned it's affecting all the other strains, right? These are the biggest influencers.
Yeah, they're the orchestrator of the microbiome. That's what we're seeing in our studies. We over and over again, we see that these strains, once put in, completely modulate the microbiome to create more diversity, more robustness, higher levels of these keystone strains that protect against disease. They bring down ammonia, they acidify the microbiome, they increase short chain fatty acid production. All of these things that are characteristic of a healthy functioning microbiome are being reset by using the spores. So all of these things that we're exposed to, all of the pesticides and herbicides and chemicals in our foods and antibiotics and all the things that work against those features are being fixed and repaired when you add the spores in.
Is there a place where we would find those specific strains in nature? Would those be in the dirt, or?
They would, yeah. They're ubiquitous in the environment. So if you go the mountain you're talking about, if you go and look at the dirt, you'll find them in there. You'll find them in on the surfaces of fruits and vegetables that you can pick naturally that's growing wild. In rivers and streams, you'll find them. So they're ubiquitous in the environment.
Okay, awesome. And then last thing, did you formulate the K2 formula? Because that was the K27?
Yeah, so K27 has been one of my passions for over 15 years now.
Oh,
and
wow.
the way I got into K27 is because it's one of the byproducts of bacillus fermentation. So these bacillus endospores make K27. That's the highest natural amount of K27 you can find is on natto, which is a Japanese food that is fermented by bacillus, right? So in one of the first projects I was working on in the late 90s, early 2000s, which is an enzyme called nattokinase, I was doing a couple of clinical trials on it. During the nattokinase fermentation and extraction, the company that was making it was throwing out the vitamin K2. And, to me, everything that is created in a fermentation soup is valuable. And I honed in on the K2 and was like, this is such a unique vitamin that we've got to work with this more and study this more. So I've been working and studying with K2 for a long time, and it's one of the most valuable micronutrients. And we get none of it in the Western diet. It's absolutely critical for heart health, for bone health, for neurological function, for hormone function, for immune function. It's an anti-inflammatory. There's nothing else from a micronutrient standpoint that does quite as much as K2 that I've seen, and we get none of it in our diet, zero. So it becomes one of the quintessential supplements. And then the dosing is really important as well. So there is a linear dose relationship with the effect of K2. The higher the dose up to a certain point, the better and more complete the effect. And so I didn't find any product on the market that had a 320 microgram dose, which is the dose that we found where you get all of the important benefits of vitamin K2. And so we created that formula with some of the supportive nutrients that are required for K2 function in the body as well.
I love it. Yeah, I'm sure you're familiar with Weston A. Price, Weston A. Price Foundation, the dentist.
Yeah, I just spoke at their show this last year in November. And I'm speaking again this year at the Weston A. Price Foundation show.
Oh, that's awesome. Yeah, because when I was vegan for years, like the biggest problem with veganism, besides the hormones and the stomach acid, to me is the dental, because it's just hell to go through cavities and having teeth pulled. And I just see it over and over. I think it's inevitable that vegans are going to have dental issues. And
absolutely.
I remember when I was vegan, it's like the third year in, and I just chipped my tooth so easily. And I think it was a K2 deficiency. So I think especially for vegans, even for vegetarians, or for people that just aren't doing raw dairy, I would say that supplement's super important.
Absolutely. Critical.
Yeah. Awesome, Karan. Well, I'll let you run. And thanks so much for coming on the show. I really appreciate it. That was awesome.
Yeah, thank you for having me. It was a pleasure, and happy to do it again if we have more to talk about, which I'm sure we do.
Definitely. Yeah, there's so many rabbit holes there I wanted to go into more. So next time.
Take care.
All right, you too, Karan. Thanks. Wow. What do you guys think about that? Really, really fascinating stuff. Really interesting that Bacillus indicus makes 12 different carotenoids in the gut, and it's intelligent. It's the perfect amount that we need. We don't need to be supplementing carotenoids, which is actually causing a lot of damage in people. So definitely check out Just Thrive. Check out their products. They just released a really cool prebiotic product. But my favorite by far is their spore-based probiotic, and I take at least one a day, usually with dinner, and I did definitely feel a shift. When you think about the digestion and assimilation of our food and the production of our neurotransmitters, dopamine, serotonin, and really the really important roles that these probiotic bacteria make, especially the influencers, as he called them, really start to connect dots and see how a lot of our health issues might be a lot simpler than we think. Just getting our gut back on track and not getting confused and trying to take every strain out there and trying to get a super high CFU count, but just taking the most important strains just once a day, and what a difference that can make. I consider it one of my top 5 supplements that I take, and maybe someday I'll do a show on the top things that I recommend. So you can check them out at thriveprobiotic.com. I'll put a link below, and if you purchase through my link, it supports my work, and you could also use the discount code Blackburn to save 10% on any of their products. You can also support me by going to matt-blackburn.com and find all my recommended products there, which I update weekly, and mitolife.co, where that's my personal brand of products that I personally sell and believe in. So that is being upgraded as well, and I have a lot of exciting products coming out soon. Today's quote is by Stephen Jay Gould. The world is full of signals that we don't perceive. Tiny creatures live in a different world of unfamiliar forces. Many animals of our scale greatly exceed our range of perception for sensations familiar to us. What an imperceptive lot we are, surrounded by so much, so fascinating and so real that we do not see, hear, smell, touch, or taste in nature, yet so gullible and so seduced by claims for novel power that we mistake the tricks of mediocre magicians for glimpses of a psychic world beyond our ken. The paranormal may be a fantasy. It is certainly a haven for charlatans. But parahuman powers of perception lie about us in birds, bees, and bacteria.